Thank you very much. What an amazing build-up. I've never had such a fantastic build-up. I'm sure I'm not worthy of that. I'm going to tell you tonight how the Royal Free is and will continue to change the world. I mean, absolutely seriously change the world. Some of the stuff that we have done, that we are doing, that we will do, they are not doing anywhere else in the world. So that is what I'm going to tell you about tonight. I'm also going to tell you a little story of 3 Frankensteins as well. And I'll take you through that. Very topical at the moment, isn't it? Lots of Frankensteins in the cinema and so on. Now, to avoid confusion, I'm a professor at UCL, and I work at the Royal National Throat, Nose and Ear Hospital. When I arrived, it was part of the Royal Free. But it's actually transferred for various logistic reasons to UCL management. However, I still feel part of the Royal Free. I want to emphasise that, and all my research is based here. So that's what I'm going to be talking about tonight. You will have seen this, I hope, on the flyer. This is a painting. It's on the wall just down here, just down the corridor down here. There are loads of fantastic pieces of art at the Royal Free, and the Royal Free Art Collection actually is very big, and if you've not had a talk about it, I think it's overdue, because it's really very impressive. And this one's by an artist called John Greenwood. You probably don't like it very much, but I really do, because I think it really conveys the idea of creating organs out of nothing. And this is his style of painting, but it's absolutely brilliant. This is supplied by Paintings in Hospitals, by this particular one. And it's a wonderful encapsulation of creation. If you do like email, this is my email address, so feel free to send me any comments that you haven't had time or embarrassed to ask or anything. It doesn't really matter. Please do email me afterwards. So I kind of got to this point by complete serendipity, and also by happening across brilliant people along the way who seemed to not be too irritated by me, which was quite good. It doesn't always happen. It's a bit like, you know, a small river starting at the top of a mountain and gathering a bit of water as it goes downhill. Can't really predict where it's going to go. It's going to be guided by the rocks that it hits and the boulders, and you know, and eventually, hopefully, ends up in a magnificent river and really contributing to the life and growth of the world around it. And I guess my life was a bit like that. It was very random. I was the world's worst medical student, and by modern standards, I would have been thrown out of university. There is no question. For academic reasons, actually, not for anything illicit. But anyway, happily, I did scrape through and ended up in ENT, doing an ENT job, my first clinical job. Not because that's what I wanted to do, because that was all that was left. And in those days, ENT was really viewed as something not really worthy of science. It was merely a means for surgeons of a certain predisposition to take out private tonsils to replace the tyres on their Bentleys. That was mainly what I thought. But as soon as I started in ENT, I realised that there was a whole vista of research that needed to be done, that it was a completely open field, from tiny little ear surgery to massive head and neck reconstruction for cancer, and that virtually none of it had an evidence base. And so I decided that this really was such an open field, I really had to go into this and try and change things a bit. So I went through my training, and I ended up in Australia in the end, because I did a lot of research in my training, and one thing that's very important for academic surgeons particularly is still to be credible surgeons. So to know about the science, but also to be credible surgeons as well. And in Brisbane, they had the biggest head and neck cancer unit in the world at that time, and I was able to do lots and lots of cutting, and therefore be confident in myself as a surgeon, as well as a scientist. And during that time, a very important thing happened, and possibly the most important thing about research, and that is getting the right research question. And the research question here was, how can we rebuild people better than we can at the moment? We have lots of ways we can rebuild people, and a lot of them are absolutely fine for their purpose, but they're very limited. We can take skin grafts, we can borrow bits of the body, we can swing quite sophisticated bits of tissue, for example the forearm on its blood vessels, and put it up here to replace the skin. You can take a bit of the leg on the fibula, which is the bone in the leg, split it and make a jawbone, and put that up here too. So we're very good at doing that kind of thing. You can use prostheses for some purposes, and in facial replacement, we sometimes work with Madame Tussauds to create very spectacular facial prostheses, but they never look quite right. They're never quite there. And the other thing you can do is conventional organ transplantation as well. So So but my research question, as I say, was how can we replace function? And I'm particularly interested in laryngectomies. These are people who've had cancer of the larynx and have their voice box removed. It means they can't talk properly, they can't swallow properly, they look a bit, they don't look normal either, and they lose their confidence, they have a high degree of depression. It really is a kind of dehumanising condition to remove the larynx in many cases. I'm now patron and medical director of the National Association of Laryngectomies and helping rehabilitate these people. But I thought, this was the question. I want to be able to rebuild these people. So here's Frankenstein number 1. So I kind of then turned into Baron Frankenstein, really, because I wanted to rebuild people. Now, Frankenstein 1 is the historic figure of Frankenstein, the one from the Mary Shelley book, which, as you may be aware, is actually based on a really ancient myth of Prometheus. Prometheus was the god who was thrown out for giving fire to mankind and therefore making people almost as powerful as gods. In fact, the gods were so angry with him that they chained him to a rock for eternity. And for every day for eternity, his liver was pecked out, but then regenerated overnight, only to be pecked out again the following day. So Prometheus, unfortunately, was really punished for having given fire to mankind, but really was the first, I suppose, god, to have actually regenerated. The other thing, the second Promethean myth, is closer to the Frankenstein myth, and it's the one that Mary Shelley really based it on, and that was that he actually created people out of clay. He animated them. Animar means soul. He actually put the soul and the heart into something of clay and made it live again. Every time an organ is transplanted, that's what happens. You take something that's dead and make it live again. Every time a transplant occurs, it's a tiny miracle. It's a tiny Promethean myth. It's a tiny piece of Frankenstein. Now, the Frankenstein that was built in the story was based on the best of technology available at the time Mary Shelley was writing, and the best of scientific theories at that time, and particularly the idea that electricity could transform all everything. Electricity was the answer to everything, and that if you had enough of it, you could actually make things that were dead live again. And that's kind of what it was based on. You take dead things, put enough electricity through, and away it would go. Maybe, by the way, in 50 or 100 years' time, people look back at what I'm saying today and they'll think exactly the same about that science as the science that I'm talking about today. But I don't think so. So conventional transplantation, as I say, is making the dead live again. But it is limited. These are the organs that we can transplant today. Lungs, hearts, kidneys, livers, pancreas, all kinds of things. However, it's limited for all sorts of reasons. Firstly, because there aren't enough donors. And anybody who isn't an organ donor, and I do urge you to sign up for that. We don't have enough organs. So most heart transplant patients still die on the waiting list, as do most liver transplant patients. So there is a big need for an improved way of supplying organs and tissues. The second reason is that if you put a new organ into somebody that's come from somebody else, you have to immunosuppress that person. You have to persuade their body not to reject it. Unfortunately, the human body, and indeed all mammalian organisms, have not one but 10 different layers of ways of rejecting things that aren't self. And this is how we actually sustain life. We have to do that in order to get rid of microorganisms, get rid of all kinds of things, including tumours, by the way, which express aberrant antigens. So it's a safety mechanism, but it's one that's incredibly difficult to overcome in order to make transplantation safe. The other thing about transplantation is that it's limited. There's a limit to the kinds of organs and tissues that you can actually transplant. For technical reasons, we can't transplant everything. So transplantation is fantastic. And since it was invented by a guy who started out as a head and neck surgeon, incidentally, a long time ago, it has saved millions of lives. But we need to go further than that. The Royal Free is, of course, the transplant centre for UCL partners. Now, we thought that we could build on this, and that this too might be the answer. And there's a scientist at UCL who spent his entire life studying these. You know what this is? It's a newt, actually. A bit salamandery, but it's a newt. And newts and salamanders do much the same thing. What happens if you cut a leg off a newt? It grows back again. You can even cut the whole lower jaw with the tongue and the teeth and the muscles and everything off, and the whole lower jaw grows back with everything in it. It's absolutely spectacular. So this guy spent his entire life studying what genes are present in newts and salamanders that make them regenerate like that. Because wouldn't it be wonderful if a poor soldier coming back from Afghanistan had a limb blown off, if we could just change the genes and it would just grow back again. He says he spent his entire life looking for the Newt within. Sadly, having got to the end of that period, that whole list, 34 years, he has come to the conclusion that actually our genome is so different from that of newts and salamanders that we can't really recapitulate that. We've gone down a different path. For similar reasons to why we have all these layers of immune rejection, we can't regenerate, because to do that you have to have stem cells which are pluripotent that can just grow like bilio extremely quickly. And if we had those in our body, we would develop cancer all the time. And that's the reason why we can't do that. It's this dichotomy. So how else can we do it? So this is Tony Atala. Tony Atala is viewed as the father of surgical tissue engineering. This is what's called a TED Talk. And this is what my kids, I hope, listen to when they're not listening to music and on their iPods. Do you know, who knows what a TED Talk is? Okay, so these are really good. So these are encapsulated small talks, much shorter than this one, so therefore much more interesting than this one. They're about 15 minutes long, and they're by world leaders who are in science and medicine and engineering and economics and astronomy and anything you can think of. And they encapsulate their entire field in about a 15-minute interesting talk. And they're absolutely superb. And this one's about how you build organs. So I've actually stolen a lot from Tony, because he's very, very good. He started out, he's a urologist, and he started out building bladders for babies who were born without bladders. And he started this work in the early 1990s. There's this thing that the Isaac Newton phrase that, you know, if I've built anything, it's by standing on the shoulders of giants. And actually that was borrowed itself from some Greek philosophers who said the same thing. Well, likewise, anything that we've done, we've built on the people who've gone before, including Tony Atala and the people before him. It takes about 20 years from a basic idea to actually get something into clinic. That's if you're successful. And only 1 in 10,000 of those ideas actually make it, in fact. But by building on what he's done, we believe we can do better. So how do you build something from scratch that's actually going to work in the body? Well, these are the principles of tissue engineering. You need appropriate cells. You need appropriate stimuli. And the stimulus can be molecules to persuade the cells to do what you want them to do. They can be mechanical stimuli too. If you take a stem cell and put it on a very hard surface, it'll want to turn into a bone cell. If you put it on a slightly softer surface, it will want to turn into a cartilage cell. And something that's even softer, it turns into a fibroblast, the cells that make most of the tissues in our body. If you put it in an environment that's actually moving in and out, it'll actually become a muscle cell. And depending on how it moves in and out, it'll become a different type of muscle cell, either a heart one or one that you might want to put into a leg. So we can persuade cells to do all kinds of things now. To put these into the body and to make them function and function in groups, you have to put them into a scaffold. And scaffolds are of 3 sorts. Firstly, you can create one out of synthetic materials, and I'll come back to that because we're very good at doing that here at the Royal Free, extremely good at it. You can put it into biologic scaffolds, and by and large these are bits of human body or animal bodies that we have worked on and stripped out cells. So we take actually human donor organs that aren't being used for transplantation, we put them through certain processes which we're now patenting, which strips out the donor cells, and that means that they don't reject. You can put these scaffolds into any body, and because they haven't got those bits of cells which tell your body that it came from somebody else's body, it won't reject. And you can then put your own cells onto this scaffold and create something new for you. The third type of scaffold is a gel environment. So these are more flexible things. These are ways of delivering cells really into what we call an extracellular matrix of their own making. All cells will try and secrete an environment around them, some cells more than others. And if you put them in a right kind of gel, they will make the kind of environment that they like. And you can also make gels which will allow you to inject things. So you can either build a surface, so you can make new skin, for example, out of a gel, or you can make it into an injectable form, for example, to inject into the heart, to repair damaged heart tissue after a heart attack. So those are the 3 kinds of environments you can put them in. You need cells, stimuli, and you need a medium. After Australia, I was really intent on the idea of replacing a larynx by transplantation. And so by support from the Wellcome Trust and from others, we worked for many, many years on the idea of conventional transplantation of the larynx. It took us a long time to work out where we were going to put the nerves and what kind of immunosuppression we would use and how we would do it. In 1998, the world's first laryngeal transplant was done in the Cleveland Clinic. Unfortunately, that rejected last year and had to be taken out. But nonetheless, that was the longest that any first transplant had ever survived. In 2010, with a group in California, we had the opportunity to do the world's second laryngeal transplant. This was different because by this stage, we know how to repair the nerves. And so to this lady, who is Brenda Jensen, this lady had not spoken for 10 years. Her larynx was completely useless. It had been scarred up as a result of a traumatic intubation to save her life. And subsequently, she'd gone on to have a kidney pancreas transplant. So not only did she have no really good larynx to work with anymore, but also she was already immunosuppressed. That got us around the ethical implications of doing a transplant for quality of life purposes rather than quantity of life purposes. And so in May 2010, we did the world's second laryngeal transplant, and she's functioning very well indeed. She has a normal voice, and you can see her on the internet if you wish. She has a normal life. She looks after her daughter and her grandchildren, and she actually travels America as a motivational speaker now. So this was a major success. But to do this, we had to find a very, very unusual patient. This is not something that we can wield out to everybody at all. She was very unusual. And also, she was already immunosuppressed, so we didn't have to bother about the immunosuppression side. It was also technically extremely demanding. It took a very large team, an international team, because we flew people in, myself and a colleague from Italy. And the whole thing cost the University of California Davis Medical Center $1 million to do. So it was extraordinarily expensive. She said, "I don't know what the future may bring, but it sure will be better than what we left behind." And I think this is not only true for her life, but it's allegorical for where we're going with tissue engineering, because the world, I think, will leave conventional transplantation behind within the next 30 years. Based on Tony Atala's work, and in parallel with working on transplantation, we started to work towards tissue engineering for larynx and trachea, windpipes. And in 2008, we were faced with this young lady from Colombia who had tuberculosis, and her main bronchus and into her windpipe was scarred up irretrievably as a result of the TB. She was getting more and more pneumonias, and she was going to die. And so, having done a lot of work on pigs and demonstrated proof of principle that we could tissue engineer a trachea using some of Tony Atala's techniques, we built her a trachea. We put it in, and she was out of hospital in 10 days, breathing normally. The 5-year follow-up paper was published in The Lancet last month, and she's still looking after her kids, and she works full-time. So this was the first time that anybody's own stem cells had been used to build an organ, and it was proof of principle. And people thought it was really exciting. I think it's exciting. I hope you do too. And in fact, this great bastion of science, the Daily Mail, in fact said that we'd heralded a new dawn for medical science, which is very nice of them, actually. The trouble is, this was a one-off, and we realised, having done it, how we'd really thrown together a lot of things and how we really didn't quite understand how it had worked. Serendipity had stepped in a big way. We'd taken bits of things which a priori we thought would work, built something, and it kind of worked, which was great. But we knew we could do better than this, so we went back to the laboratories. I moved from Bristol, where I was at the time, to here, because I knew this was a much, much better place to build these technologies and make things better. So, for example, we realised that the scaffolds we were putting in were too floppy. So we spent years and years perfecting the right scaffold. When you strip all these cells out, they go a bit soggy. We now know how to retain the biomechanical rigidity to make it just like the tissue and organ that you took out in the first place. And it's uncanny. When you handle one of these things, it's exactly like handling a native organ. So we're very, very good at that now. I've had a whole series of clinical fellows and PhD students who've done all this work. I haven't done anything, by the way. I just stand around and talk about it. It's fantastic. You have all these bright people. You employ enough bright people and you get good results, and it really works. This is Allen Partington, who has just got her PhD and has now moved off to the cell therapy catapult to actually create even better organs than this. So we worked and we worked and we got better organs and tissues. And in 2010, we were presented with a real challenge. This is a little boy at Great Ormond Street who had been born with a really narrow trachea. And the only way that he'd been kept alive over the years was by putting in a metal stent. A stent is a tube which holds open a hollow structure which has been damaged. Unfortunately, over time, these metal stents erode. They move out from where they're supposed to be. And this one eroded into the main blood vessel in his chest, the aorta. He woke up one morning, coughed, and blood started coming out of his mouth. Now, he was actually stabilised at Belfast Hospital, this Northern Irish lad, and flown down to Great Ormond Street. And the Great Ormond Street team said to us, we can't do anything here. We can replace the aorta, but we can't replace the trachea. Can we do what you did with Claudia Castillo in Spain? So we had to, again, put together a protocol which was much, much quicker than the one in Spain. We reduced it from 3 months down to a month. We had an on-the-shelf decellularized scaffold which we could use, and we used his own bone marrow stem cells, which were taken at Great Ormond Street and then couriered up to the Royal Free Laboratories, the Laboratories of Cellular Therapy just over here, which is the country's leading centre for cellular therapy. By the way, I digress a little, but I will advertise a bit more. I don't mind. I'm allowed to advertise the Royal Free, aren't I? OK. So in 2011, 2012, the Royal Free alone delivered more cell therapies, both experimental and therapeutic, than the entire of France and Germany combined. And we actually deliver more clinical trial cell therapies than the entire of the rest of Europe. Isn't that absolutely incredible? And one more statistic, actually, just in case I forget for later. There have only been 6 successful gene therapies in the world. Gene therapy is part of regenerative medicine, by the way. Only 6 successful gene therapies have made it through phase 1-2 clinical trials. And 50% of them are at UCL. And one of them is here. So how about that? That is how good we are. We are really that good. Anyway, so we put all this together and we put this trachea back into this boy. And I'm pleased to say that he is very much alive and well today. In fact, he's so alive and well that he won... Oh, hang on. Come on, Kieran. Where are you? There he is. This is Kieran. He won an egg and spoon race recently. We actually had him back into hospital because we didn't replace everything. There's still a little bit of bronchus that's a bit naughty, and we have to clean from time to time. And his trachea looks completely normal. It's got normal epithelium. All the biopsies are completely normal. So this was the world's second stem cell-based organ transplant and the first stem cell-based organ ever to be put into a child. And he's doing really, really well. He played the drums on Irish national television at Christmas. Okay, but still, we still know that that wasn't perfect. He was actually in hospital for 6 months afterwards while we cleaned the trachea and kept it clean and kept supporting things and putting new stents in. And then it firmed up and then we were able to discharge him. We need to do much, much better than that. Now, one way we can do better than that is if we didn't have to rely on donor organs, if we can actually make things out of synthetic biomaterials. And on the top floor here is Alex Seifelian, who's the country's leading nanotechnologist. And he has developed the most amazing material over the last... He started out as a nuclear physicist in Iran, incidentally, but found that Britain was a much better place in which to develop material science. He's an absolutely brilliant guy. And so this material, which he developed over a period of 20 years, has now been used to rebuild a whole load of organs, which I'll show you a bit later on. It's called POS-PCU, and it's patented to UCL. In 2010... end of 2010, my PhD student created a trachea out of this biomaterial, which was put into a man from Iceland who was at the Karolinska Clinic in Sweden. And this is the only time that a synthetic trachea has ever worked. People have been trying to build synthetic tracheas since 1952. And they've all failed at the 3-month mark because synthetic materials don't really integrate into infected areas very well. That this area is all colonies of bacteria and synthetic materials don't mix. This is the only time this has ever worked. Okay. But these are all what we call case reports in medicine. These are all one-offs. Every time we've done one of these, they've all been very different from the one before. This is Chevalier Jackson, who was one of the pioneers of surgery. He's an American guy, and he came to address the Royal Society of Medicine. And he was saying how you can learn a lot from case reports, but you have to be very guarded about what you learn from them. They often make a big media splash. But what you really need is to go back and study what you did and try and understand what the underlying science really is and where you need to work with scientists to make things better and better. Furthermore, you need 2 other things. You need more than one. One swallow doth not a summer make. You need more than one. You need to do clinical trials, proper clinical trials where everything's measured and we have external monitoring and you have data committees who can say, you've done too many people have died, you need to stop this right now. Proper clinical trials with numbers of patients. And you also need time. Tony Atala says you need to wait 4 years before you know whether one of these things works or not. And he's probably about right. So we now know that Claudia's trachea has lasted 5 years and Kieran's lasted 4 years, but we need to do an awful lot more. Didn't used to be like that. This is Donald Thompson, and he won the Nobel Prize in 1990 because he invented bone marrow transplantation. But when he started, his first patient that he transplanted died, and his second one died, and the first 50 died. But each one of them lived a tiny bit longer than the one before. And I think it was actually the 53rd, not the 50th. The 53rd patient survived. And this has now saved millions and millions of lives around the world. Bone marrow transplantation is a real lifesaver. But if he was working in the modern America with the FDA, their regulatory agency, he simply wouldn't have been able to do that. If he was working in Britain, he would have found it very hard. But happily, the regulators here have a much more rational view of progress and a much more balanced view. And you can go and talk to them, and we do go and talk to them very regularly. The MHRA actually is growing at the same time as this. Because the regulators, they've been invented to protect the population. And most of their methods are based on making drugs safe. And the method of taking a drug from the bench to the clinic is very well established. Not so when you're dealing with something which involves a bit of tissue and cells and maybe stem cells and maybe even gene therapies as well. This is a completely new world for regulators just as much as it is for us. Happily, in Britain, we are growing with our regulators at a rate that America is not. And so we actually still have a competitive advantage. But we're constantly reminded that we still need to keep thinking about the science at the same time. This was a science article that came out earlier on. This is the world's leading scientific journal. And this was actually highly critical of what we were doing. And I'm glad it was critical. And what it said was, basically, we're going into people too soon. We need to understand everything about everything before we go into man. Well, if we did that, we probably wouldn't get there. They were working on heart transplantation in Boston for 20 years when Christian Barnard rocked up to do a 3-month fellowship. And they were saying, well, you know, we've nearly got it, but we're not 100% certain this is going to work. So we need to do this extra experiment, and this extra experiment, and this extra experiment. Well, he said, well, actually, I'm pretty sure this is going to work. He took it back to South Africa, did it, and heart transplantation is what it is today. Somewhere, there is a balance between the two. We can work with the scientists to make things better, and some of the scientists working in this hospital and at UCL are absolutely brilliant at this. This did us a huge favour because it actually has enabled us to go back to the funding bodies to say, look, science says we need to do this, this and this. Please, can we have another £2 million? So actually, I'm very grateful. And they're right. But that was actually driven by a political thing in the States at the moment, whereby Obama has put a lot more money into translational research, that is taking things into people, than he has into basic science research. He's cut the NIH budget for basic science. And so the basic science are trying to claw it all back. They don't need to do that. They simply need to talk to us because we can tell them a lot of things about the body and give them ideas about developing basic science. We're trying to make things better. One of the things we're making better is building mucous membrane in the laboratory to try and line the synthetic tracheas that Alex is making and try and line our grafts as well. So we're trying to work out where is the epithelial stem cell in the mucous membrane. And I have very young scientists who's working on this, who's making some really big breakthroughs. And we're trying to use that to rebuild larynxes now. And this is a larynx of a pig which we rebuilt recently. And if you're interested, if you rebuild the larynx of a pig, it still grunts. And there's actually a BBC podcast where we talked about our pigs, and you can hear the pigs grunting before and afterwards. And somebody said it went from sounding like an opera singer to sounding like Barry White, but I haven't heard many pigs that sound like opera singers or Barry White, really. It's quite impressive. Based on this, one of our first products that we're going to be able to produce here at Royal Free is a mucous membrane replacement. At the moment, if you want to replace mucous membrane in head and neck reconstruction or into the esophagus, you have to use split skin grafts, which really aren't that great. Anybody who's been in the bath for too long realises that your skin goes a bit soggy and a bit sad. And that happens if it's exposed to mucus in the throat all the time. So we're now building mucous membrane, and actually we can do this. This is actually Sheffield technology, but we're moving it down here. We've stolen the scientist responsible who's going to be starting at the Royal Free in January, Anthony Bullock, and he's going to be building really nice bits of mucous membrane, which hopefully we can not just use for the head and neck, but we can use for esophagus, we can use for bowel, we can use for waterworks, all kinds of things. We're also looking at rebuilding esophagus. We work very closely with Great Ormond Street. Great Ormond Street don't have the cell therapy laboratories and what's called GMP, that is clinical grade laboratories, that we have here, and therefore they rely very much on our clinical grade technology at the Royal Free to deliver any of this kind of work. So some babies are born without enough esophagus. This is called a tracheoesophageal fistula. And one of the great things for tissue engineering here is that you actually don't have to build very much. These are very small babies. You only have to build like a centimetre of tissue. This is a very realistic goal. And we've just had a £1 million grant from the UK Stem Cell Foundation to start doing exactly this. And we hope we'll be able to start transplanting our first baby within the next 2.5 years. Okay, that's more esophagus. This is not our work, this is a lung, but it's just to show you the diversity of things that are going on in the world. This is a group in Boston who used exactly the same techniques that we did. They didn't give us any acknowledgement. I don't mind, so long as it helps people, that's absolutely fine. But of course America's much better than Britain, apparently. There we are. So they took a lung in a rat, they stripped out the cells, they put in some new stem cells, and then they put it back into the rat, and it ventilated that rat for 8 hours, gave it enough oxygen for 8 hours. So maybe lung transplantation is round the corner with these things. But blood vessels are a lot easier. You can put in a bit of, Blue Peter. They used to build things out of squeezy bottles in Blue Peter, old John Noakes. Well, you can put in a squeezy bottle, and it would still probably function as a blood vessel for a bit, because there are circulating stem cells in the blood which will line it. Nonetheless, we still haven't got the ideal replacement for blood vessels. And this is some work from America which shows that the stem cells will actually grow on the inside. So this here is the work of Doris Taylor from Texas. And she has shown that you can take a heart and you can strip out the cells and you can grow some muscle cells in the laboratory using the techniques I was telling you earlier. Remember, if you stretch them and shorten them, you can make them into heart cells. You can then put them onto a heart scaffold. This is a rat heart scaffold, and it starts beating. And it actually has a cardiac output, a very, very small cardiac output, but it does have one. But what are we doing here? This is George Hamilton. He's the director of surgery here, and he's a brilliant surgeon scientist as well. And working with Alex Seifelian, he's been able to develop vascular grafts based on post-PCU, that wonder material I was showing you earlier, which we've invented here. And here he's showing you it's a bypass graft, and Alex and George have got a very large grant from the Wellcome Trust to do a randomised controlled trial of using post-PCU as coronary artery bypass grafts. And that will start recruiting next year. And it will hopefully replace the need to take blood vessels out of the leg or from the inside of the chest. It'll be very much more reliable. Apart from anything else, you can totally personalise these. You can actually use the same technology that's used in inkjet printers to print these to the exact shape and size of your coronary arteries and put your coronary arteries back into you made out of post-PCU. And that's what they're going to do. Absolutely brilliant man. Alex is also working with the cardiac surgeons down at the Heart Hospital to build heart valves out of the same material. And likewise, he's had a very large grant recently, and these heart valves will also be going in within the next 2.5 years. Isn't it great here? This is all happening here. This isn't happening here, but I sincerely hope it will. This is a kidney. And a group in America recently built a kidney that worked in a rat. They don't say for how long, and they don't say what the urine was like, but they did manage to make it work. And I can't tell you who it is, but we are hoping to headhunt and recruit a guy from the States, who actually is from Italy originally. And we hope he will actually set up potentially a kidney regeneration program here. And what better place to do it when surrounded by this mass of people working in other fields, but also in London's centre for the kidney. This is the liver. We're also the centre for the liver as well. And liver transplantation is very problematic indeed. This is Brian Davison, who's another one of our brilliant surgeons here. He has already built a kind of liver that works outside the body. And so for people who are waiting for liver transplants, remember half of them die on the waiting list, as I told you earlier, those people can be actually tided over while they're waiting using Brian's invention of a synthetic kidney for outside the body. It's not yet in clinical use, but it's there, and it will be very shortly, I'm sure. However, there is an international challenge to be the first team in the world to transplant a fully tissue-engineered liver. Not into humans, they've been a bit realistic, but into a large animal of some sort. And so now the race is on for us to try and beat groups in Japan and America to this £1 million prize, and be the first people to actually do that. And we've got all the bits and pieces to do it. So the race is on to do that. This is Alex Seifelian, who I was talking about earlier, and this is his printing machine. So here he's printing a windpipe, and looking absolutely amazed that he's actually done it. But he has actually done it. Again, I was hoping to actually bring all these down here, but I've been so useless in finishing clinic, I haven't managed to do it. But if at some point in your wanderings you wish to wander up to the 9th floor, Alex will be very happy to show you around. You don't even need to go into the division of surgery, because just outside is this cabinet. And this cabinet has within it examples of all the organs and tissues that he's making out of post-PCU. And it's really staggering, the breadth of things he's been able to do. And this is an ear. I'm an ENT surgeon, although actually I'm really a T surgeon because I only do throats. But historically I'm an ENT surgeon, and I work at the Ear Institute. And so we've actually developed a pan-UCL collaboration now to engineer the best ears. At the moment, if you want to rebuild an ear that's been lost due to tumour or trauma or burns, or a child is born without an ear, you have to undergo a very complex process of reconstruction using rib grafts, which don't always work, or implanting a German material called Medpor, which sometimes extrudes and gets infected. Post-PCU doesn't do that. It's really biocompatible. It stays in. So you can spin ears to the right size and shape, and you can implant them. And Alex, along with this man, Peter Butler, who's Professor of Plastic Surgery here, hope to be starting a clinical trial of this in India in the next couple of years. They've already got... India has a really big problem with burns and trauma and tumours of the ears, and so have a very big population. So working with plastic surgeons in India actually makes a lot of sense, in fact, to do this kind of work. We're also working with the surgeons at Great Ormond Street who rebuild the ears for children, and we have ways of using a particular type of stem cell called amniotic fluid stem cells, even to build ears for children before they've been born. If you can identify a child in utero as not having external ears, we are now able to take cells from the fluid around the baby, which is absolutely full of their own stem cells, and use that to create an ear, or any other organ theoretically, for the baby before it's even born. This is a nose. We're also building noses as well. Okay. But none of these things move apart from the heart, of course. Wouldn't it be great, going back to the surgeon who lost his limb and going back to the Newt within, if we could actually build things that actually move, arms and legs? This is a robotic hand. Robotic limbs at the moment are a bit clunky. They do work. We are able to get people walking again, and we are able to get people picking things up, and they're getting better and better. But you're still reliant on kind of almost Victorian levers and pulleys and joints to make them work. So we're working with the Bristol Robotic Centre, which is the biggest robotics unit in Europe, on something which we think will do the job rather better. So I hope this runs. Okay, so this is what we called electroactive polymer. Electroactive polymer effectively is synthetic muscle. This is a material, this is an eyeball being moved around, so this is if somebody had an eye removed, then you can actually put muscles in which work. You can actually make this deform in a very predictable manner by applying a voltage across it. Now at the moment, this voltage is a bit high, but that's coming down all the time. That's problem number one. Problem number two is coating these materials in a way that the body will accept. And that is what we're most closely engaged with right now. So the Bristol Robotics Laboratory is sending us their materials, and we're coating them in our miracle post-PCU so that the body will accept them. And in that way, we think we'll be able to make, to start with, very little muscles, but muscles nonetheless. Things that might make the face move in children who have facial paralysis. Things that might make the eyeball move around. Things that might make, from my point of view, very selfishly, voice boxes talk again. I think we can do that. Good. Okay. Sorry, I was going to moan about the amount of paperwork involved, but it's okay. It's not a night for paperwork. I'm not going to talk to you about it. I'm not even going to talk to you about this, really, which is the regulatory pathway that we have to negotiate. Because, like I say, I actually sympathise with these guys, and there is a fast-track way through all that. However, I can moan a little bit about this. This is called the Valley of Death. And this is a very well-known phenomenon, and it's the gap between basic research and clinical research. Now, the problem here is that funding bodies, the conventional funding bodies, give out money on the basis of high-quality science, and absolutely right. And we are right at the top of the world in terms of science in the UK. And in fact, UCL has now moved up. It's now 4th in the world in terms of science. That's extremely good indeed. It's phenomenally good. And Cambridge, which is 2 above us, is 2nd in the world. So we've got 2 out of the top 4, which is pretty good. Nonetheless, we've done that on the basis of excellent basic science. To get something actually that we know works in an animal into the first clinical trial does not require really clever basic science. It requires a lot of legwork. All that paperwork you saw in the previous document. It requires doing things over and over and over again, in the same way as you would if you were developing a new car or a new tyre or a new aeroplane. Over and over and over again, because you have to do that to demonstrate that what you're producing is going to be the same every time. That you're not going to put something totally different into different people. That you actually understand what you're putting in. That you've got all the processes lined up. That you can do it in a sterile way that's going to be accepted by the body when you produce it. That what you're doing is actually going to be cost effective as well. That it's not going to cost £10 million to create a small bit of skin. Like the guy, you know the guy who made the steak? Did you see that? There's a guy who actually made a steak from stem cells and ate it. And that, it cost £10,000 per steak. Well, I'm not going to have those steaks in the near future, I have to say. But that's just an example of you know, how you've got to make things balance up cost-wise. Now, in the past for drugs, this gap was filled by drug companies, but the pharmaceutical industry is no longer able to step in. This is also a new area. They're very tentative about stepping into regenerative medicine because they're not absolutely certain how much income they're going to get back again. And so into this space has stepped very, very few funding bodies. The MRC and DOH do have a pot for this kind of stuff, but largely this has to be bridged by charitable funding and by the support from angel investors, by setting up spin-out companies and raising money in these various ways to get it across this gap. It's very, very expensive. In fact, in this country it costs about £1 million to get across that gap for any product. In the States it costs 20 times that much because the cost of healthcare and science is so much over there. So, nonetheless, we are not daunted because we've already raised that much for a couple of our products and we plan to raise it for everything else. I did promise there would be 3 Frankensteins here. So this is Frankenstein 2, who actually is called Rex, disappointingly. I'd rather have hoped he would be called Frank, but never mind. And this is part of a Channel 4 programme, and this is now in the Science Museum. You can go and visit it. And the eagle-eyed of you will spot the royal free component here in that he's got one of Alex's windpipes in the centre there. So if one was to build Frankenstein right now with the best of British, this is what he would look like. Not perfect, but he kind of moves, he would breathe a bit and he could see some stuff in a kind of insect eye kind of way. So he's got limbs that sort of move around a bit. So it's a bit patchy, but it's kind of getting there. So we can sort of do a Frankenstein now. However, as you've just heard, going back to this picture which I showed you at the beginning, we're already well down the pathway of being able to replace conventional organ transplantation. I've mentioned almost all of these and some that aren't on this diagram as well. We've actually extended the range of what transplantation can actually achieve here at the Free. And so what we hope is that Frankenstein 3, Frank 3 in the future, the space guy who gets injured in 30 years' time and has to come back to Earth to be reconstructed, that he will actually end up looking like you and I, and that his organs will indeed function like a normal kidney, a normal liver, a normal heart. We really do think that that will happen. You will already, I hope, have come to this talk. It's one of the previous talks, and this was by Hans Staus, and he was talking to you about the absolutely fantastic institute that's going to be built just outside here, and the first part of which is already built. And I'm sure that if any of you are interested, then you could be shown around phase 1 of the new institute, which is just on the floor over here. And it's absolutely wonderful, state-of-the-art accommodation. And it's the old phrase, build it and they will come. So before it was built, there was some doubt about whether the scientists would move up from, you know, kind of, because it's more important down in the centre of London, isn't it? So, but as soon as it's been built, scientists have been clamouring to actually move back up here to the Free because it's so nice. Phase 2 will be in this brand new building outside. Some of the funds for which have been raised, but not all of it just yet. And at the moment, we have been penciled in to put the work I've just been talking about into one of the floors within this institute. And it sits beautifully within the Institute of Immunity Transplantation, firstly because it is the future of transplantation, and it absolutely is, and secondly because the immune questions are still very current and very important. So this is an ideal setting for it, an ideal location. We could not be better placed than we could at the Royal Free. Just as a matter of interest, as a part of this whole thing, we're also going to be putting in the operating theatre of the future. And George Hamilton is already working hard on how we're going to do that. There's a team from across UCL, and we're going to build an experimental operating theatre here, which will have the best of imaging. So the idea is that a patient can be on the table, and the imaging will come to the patient. And we'll also have robots there and lasers there. We'll be able to scan the patient. So, for example, if you were to, in my field, if you were to resect a cancer of the jaw with the skin, you could immediately do a CT scan on the table at the time of surgery. The CT scan would be turned into a three-dimensional image. This would be transmitted to a box next door, which would then spray post-PCU into the same shape as the jaw and would actually put the patient's stem cells onto this thing, which would then be transmitted immediately into theatre and plumbed into the patient. This is real sci-fi, isn't it? But these are all things which we're starting to be able to do individually. And put them all together, I believe we will be able to do all of that. And we would like to call this the UCL Centre for Applied Regenerative Science at the Royal Free. So, you'll be relieved to hear I'm at the end of all that. I have a lot of people to thank. Mark Liddell is the guy who runs the cell therapy laboratory. He's the fixer. So he is one of only 2 QPs in the country, that's qualified persons. So he's able to say that a new product can actually be put into human beings. There's only 2 in the country. The other guys is entirely private, and we're very lucky to have him here and his expertise. Otherwise, we simply wouldn't be able to get stuff into people as quickly. Alex Sofalian is the brilliant biotechnologist. Paolo De Coppi is a surgeon colleague of mine at GOS, and he is not only a fantastic and compassionate paediatric surgeon, he's actually a Nature-level publishing scientist as well. He discovered the amniotic fluid stem cell. Sam James is a Wellcome Fellow. He's a respiratory physician who has developed a possible treatment for lung cancer. Chris Mason is the editor of the Regenerative Medicine Journal. George Hamilton has spoken about. Peter Butler, brilliant surgeon. Brian Davison, brilliant surgeon. People here at the Royal Free. Also Peter Belaski and Greg Farwell, who did the transplant with me, and our funders, particularly the Royal Free Charity. I'm very glad that they sit up and take notice. And furthermore, they've not only sat up and taken notice, but they've actually funded some crucial, critical parts of this work as well. And based on that, of course, based on the amount of funds that we've been able to get from the Royal Free Charity, we've then been able to do the work to persuade these other people to give us much, much more. So you give a bit, they give more. So we currently have something in the region of £7 million based on an initial Royal Free investment of around 200,000. How's that? This was Christopher Reeve, who was, of course, Superman. He unfortunately died before he saw the first products of stem cells. But he believed strongly that they represented an important part of the future and helped set up one of the world's first stem cell institutes in Israel. And he said, so many of our dreams at first seem impossible. Then they seem improbable, and then when we summon the will, they soon become inevitable. Well, when I first started giving this talk, that everything seemed impossible. A few years ago, it all seemed improbable, but the more times I give this talk, and the more science develops, the more work goes on here at the Royal Free to completely change the future for people needing organ and tissue transplantation, the more I realise that this is actually inevitable. It's happening here, it's happening now, and we at the Royal Free are leading the world. Thank you very much.